Chris. So now we continue. It's Archit is gonna take care of this last part of this tutorial, and it's gonna also present some of new development that has been done in the group. Go ahead, Archit.
Okay, thank you, Mariano, for that section. It's very, very nice. Okay, so I'm going to talk about umbrella sampling and how that works. So, so we just heard about SMD, and we heard that it's very difficult to get free energies from SMD. And we also heard about ABF, which is an equilibrium method to get free energies. Now we're going to hear about another method known as umbrella sampling. And so the way that this method works is that essentially what you're doing is you're creating multiple windows of some molecule. So let's say that you want to know the energy of permeation of this molecule from one end to the other end. The way that umbrella sampling works is that you're running separate windows of this molecule. And so, for example, you have a window here, or you can have multiple windows throughout the protein. And you would sample each of these windows simultaneously, and you would sample them independently in this simulation. And the way that this works is, let me go back here. What you're doing is you take that location and you apply a harmonic restraint at that position. So this xi term you see right here, what it refers to is the reaction coordinate that Mariano has already discussed with you guys, you folks. And you have some position. So this molecule can essentially fluctuate within a cylinder defined at this location right here, or sorry, at this location right here, which is some position that you want it to sample along. And then you could extend this throughout multiple windows, and you can create this pathway. And then this would, by doing all this sampling, you could then derive the free energy profile from here until the end. And for the sake of simplicity, I'm not going to go into much detail on replica exchange umbrella sampling, even though that's included in this tutorial. I'll include it at the end of what I'm talking about, to give you folks a better idea. So first off, let's talk about setting up these umbrella sampling windows. And actually, I'm going to discuss this in a little bit more detail in at the end of this talk. This is actually very important. But the way that this is typically done is you will take the SMD trajectory. So the SMD that Mariano described, take that, and then you'll define the windows along this SMD. And this will give you the starting state for the umbrella sampling that you'll be doing. So here you're looking at Z from -13 until 5, which is this Z coordinate along the protein axis right here. So let me show you the scripts here, so this could make a little bit more sense on how this actually works. Give me 1 second. So let me show you how these files are supposed to look like. So first off, what you need to do to generate these to actually do this umbrella sampling is you will need the actual windows themselves. So for this problem, you should have 19 windows. So windows from 0 to 18. And these windows in this tutorial are included in this file. 1 second. They're included in this file right here. And so each of these windows, this thing that you see AMDT 0.restart.core, this is going to tell you, so let me show you here, what the position, what the initial starting point is of that molecule. And so this is where it starts its simulation. And then what you'll be doing is you will apply a restraint on that molecule at that position. So that's included in this script right here. So let me show you how this script is going to look like. So if you look at this script right here, usbase.in, and you'll see that the Colvar is similar to what Mariano had showed you earlier. It's this translocation Colvar with a distance z and a reference, as well as the nitrogen of the ammonia that will be restrained. But then you have this term right here, which is this harmonic term. And so what this is doing is essentially this is going to restrain this, this ammonia at a particular center with a particular force constant. This can be shown here. So is this making sense so far? Does anybody have any questions?
Yeah, how do you save save those windows from the DCD trajectory of SMD simulation?
Sure, yeah, that's a good question. So let me show you how to do that.
No, but isn't it very painful to just go and point out those points and then save it?
So there are scripts that can do that for you. There's scripts that can make those windows. And also, but give me 1 second. So if you go to this directory, example input, you'll see there's a get windows script. And so we have there's ways of doing this. Just using your S&D, you can do it such that all you really need is you have to figure out at this, because you want windows, let's say that are 1 angstrom apart. And so at each of those 1 angstroms, you go to your S&D and you can figure out that, okay, this, at this frame, you have something that lies within this window. So you can stitch it out that way.
So why do I need to do SMD? I can just put it with the help of a TCL script.
Yeah, so that's the thing actually. So it's it depends on how you're how you're placing these windows is actually really important. So that's an important part of actually solving this problem. Actually, that's what I'm going to talk about in a little bit more detail later. How you actually are setting up these pathways can give you actually different free energy results. Okay. But usually people use S&D because that's a connected pathway. So you know that this is a pathway that the drug could actually go through. And also the thing is that when you're doing this umbrella sampling, there's not really a guarantee that you're actually going to sample everything, every single possible position that this let's say this substrate is taking in the protein. And so this pathway, this initial pathway that you have, is actually really important. And so they use S&D because that's a sort of an easy way of talking about a connected path. But that's not always the best thing to do, which I'll talk about later. Are there any other questions? Okay, so keep going. Yeah, so let's see. Sorry. Okay, so what I was also saying is that yes, this harmonic thing, this part right here, this is basically just telling you how are you restraining at those different windows. And the force constant is actually pretty important. That can tell you how much is this thing allowed to fluctuate within that window that you have. And so this is actually really important because the amount that you're fluctuating is actually going to give you, if you fluctuate too little, then you're sort of restrained within that little position that you're like, it's like a spring basically. If you have a spring that's really stiff, that has a very high force constant, then you'll be kind of just going within that location right there. And so there won't be much overlap between these windows, which you really need when you want to build the PMF itself. But if it's too loose, then it'll be difficult to actually sample that window itself, and you might drift off into other windows. And so it's a balance between those two to generate this force constant right here. But they're saying that in this tutorial, they're saying that for a 1 angstrom window, a force constant of 2.5 kcals per mole is pretty good to get a good overlap. And here we're actually using a smaller window, which is 0.1. The width of this is 0.1 angstrom, so the force constant will be scaled down to 0.025. Okay. So is this all clear, or does anybody have any questions? Okay. So the next thing you do is, now that I've described how to actually set up the windows from SMD, and I've also described how the collected variable file should look. The next thing you do is actually to generate in each window, to generate a separate CoreVar file as well as a separate configuration file. So the way this is done is, you look at, so I'll show you both of these kind of base files. So if you look at this base file, you'll see that there's this general harmonic script. So this is not within each window. This is just an overall general file. You'll see that it'll say center right here. You look closely right there. And so what you want to do is you want to make this so that in each of your windows themselves, you have these example output, right? And so within here, you have different windows. And so you want it so that in each window, you have a different center. So let's go to window 0 right here. I can show you what I mean. You go down to the bottom. Instead of having this being centers center, this is going to be centers 5 because you're doing these windows separately when you're doing this umbrella sampling. And to make this simpler, we have a simple script that can set up these windows. So you're going to be using this setup window script right here. Before I show that, let me also show the configuration file. So in addition, you also have the configuration file, which is similar to a regular NAMD configuration file, which you've seen earlier. It's a little bit different though, because you have a couple other things you have to take care of. So one thing is this bin coordinates. This is going to tell you what coordinates should you start the system at. And if you notice here, they have this variable num. This is basically telling you that you're at a particular window. So that's going to be the num variable. This extended system is also going to tell you that you have the XSC file that you're using. That's sort of telling you like how is the system looking like is at a particular window. And you also output it with that window name. Your parameter files will be the same. And then also you have to, you have restraints, which are similar to what my analyte described. And then your CoreVars will just we'll just refer to the CoreVars file for that umbrella. And then you'll use this setup windows script. And this way you can set up it so that in each of your windows, so in each of your 19 windows, you'll have a different CoreVar file. As I said earlier, you'll have a different center. And then in your in that you'll also have a different configuration file, where you're setting the number to be 0 here. And then you can just run this umbrella sampling simply. You just do it, just running it with NAMD2 would work as normal. And so the way to run this in practice is you would run it with a bash script that would run all of your windows simultaneously at different, basically in parallel. And so this way you could save a lot of like actual time because you don't have to run everything like at once and try and sample everything. You can just kind of shoot it out all together. And you can see if you can get enough sampling that way. And after you do this, after you run all of these scripts, then you can use a program known as WAM, which I think it's not actually included. Just give me one sec. Yeah, so then after you run these each of these windows separately, then you want to construct the free energy. But before you do that, you want to make sure that your sampling is sufficient and that you have good overlap. So by overlap, I mean that the windows are like the histogram for the sampling for that collector variable overlaps with a different window. So actually don't see that the graph or the plot here, but I can just show it to you in annotation. What I mean by this. So what I mean by this is, let's say you have a plot right here. And let's say that you have, I'm just going to draw two of these collective variables. So let's say your collective variable is Z. And so by the overlap, I mean, let's say you have sampling here. Then you want it such that there's an overlap between these two, the sampling between, let's say, something that's centered here and something that's centered here should overlap. So this way you can ensure that your free energy is continuous. And then after doing this, you can, after ensuring that this is the case, then you can construct a free energy profile as shown here. I'm not going to go over all the details of how to construct this, but the way that it's done is they use a program known as WHAM, or a weighted histogram analysis method. And you can download this on what's his face? Professor Alan Grossfield's website. He has a code for running this weighted histogram analysis method to obtain these potential mean forces right here, as you can see here.
It's called loose.
Yes.
You're referring to loose?
No. He's the developer.
Oh, he has a different code. Wow. Okay. Gotcha.
Yeah. So yeah, and as Ahmad said, he also developed Loose, which is that's a little off topic, but it's a nice analysis method you can use if you want to analyze your system. So he's a pretty strong developer. Okay, so are there any questions about any of these things? If not, I can briefly describe how bias exchange works, or replica exchange works in conjunction with umbrella sampling. I'd say typically umbrella sampling, if your system is relatively simple, then umbrella sampling is usually sufficient. But if this is a problem where you're not getting good overlap, then bias exchange could really help your system. But I'm not going to go over all the nitty-gritty details because that might be a bit too complicated for this session. But just give me one sec. I'll go over the theory though. So the idea of a replica exchange, or it's also referred to as bias exchange or umbrella sampling, is that this way you can ensure a more uniform sampling. So you can ensure that your sampling is sort of continuous along the space. And the way that you're doing this is essentially, actually, let me annotate again. That really helps. Make these points clear. So the way that this is working is, let's say that you have a reaction coordinate. Let's go back to Z. And let's say that you're sampling in let's say this space. So you're sampling along this window right here. And then you also have another window, let's say, defined here. So the thing is that when you're sampling along here, you can sample a lot, and you can sample, and since you're only restraining this Z right here, then it's going to sample other degrees of freedom. So it's going to sample the angles of the substrate, or also substrate sample the XY space. But you may not actually have overlap with this sampling right here. But the way that this works is that you actually take a conformation that you have from here, and then what you're doing is you're doing a replica exchange of the parameter, or the Hamiltonian, or the restraint that you're applying on this space. You're exchanging with something here. So exchanging that parameter with the parameter of the window next to it. And so this way, this is ensuring that your space is more continuous, because now your exchanges are sort of enforcing that. So as you can see here, they're saying that changing parameters between these different replicas, you can find the conformations as well as states that could be inaccessible for another replica due to a high energy barrier along that reaction coordinate. So for example, let's say that your example is like a drug that has a certain angle, and let's say you want to sample that angle. But in this reaction coordinate, maybe sampling that angle is very costly. Then, but let's say in the next reaction coordinate, it's not as costly. What you could do is you could exchange the parameter from this space over here, and this would ensure that you're allowed to sample this this angle in this other space. Because does that make sense? And so this way you're getting a global sampling of your space. And the way that these exchanges works is it would just be it would just be using a Monte Carlo switch. So you'd have you'd make a Monte Carlo move between sampling one window and the next window. So sorry, configuration in one window and the next window, and that way you can enforce these exchanges that are occurring. And this way you can get a better a sampling of the whole space. Does anybody have any questions?
Sorry, may I ask a question?
Sure.
Okay, thank you. My question is from the previous section. When we do umbrella sampling, we have several windows. For example, when we want to pull a drug through the membrane, then we have different windows, and we define a reaction coordinate. And along that coordinate, we are pulling the drug. I want to know that, can we say that reaction coordinate is also a Colvar or not? I want to know the definition of, what's the difference between reaction coordinate and Colvar? It is also a variable that is changing in the process. I want to know the exact difference between these two. Thanks a lot.
Of course, yeah. Yeah, I think this is this terminology, even for me, it gets very confusing. I hear like Colvar, reaction coordinate. There's also what's the other one? Order parameter. It's very confusing. At least from my understanding, what it seems to mean is that Colvar can be any collection of like any collective variables. For example, like the center of mass of a Z is a collective variable because you're taking multiple variables of all the atoms, and you could say this is the Z, the center of mass of that. Whereas a reaction coordinate, as far as I understand, is something where you're actually looking at the PMF, so you're actually constructing the PMF on there. So a reaction coordinate would be a Colvar because it would follow along that sort of, I guess you could say, umbrella of Colvars, where your reaction coordinate is just what's used to actually define the PMF. Whereas the Colvar is any sort of collective variable that you have. And so Colvars is also just referring to the tool in NAMD, which is Colvars. Whereas, reaction coordinate is just what you're actually using to project onto your free energy. Did that answer your question? It's And a lot of times people will use these terms interchangeably. Reaction coordinate, CoVar, it's very, I don't think there's any sort of strict kind of thing saying that you should use reaction coordinate here, as far as I know. I don't know if anybody else can comment on that.
Thanks a lot. And another question is that, for example, when in the previous tutorial, it was said that when we want to, for example, pull ammonia through the protein, we exert potential, harmonic potential on the protein, because we don't want the protein to move and only ammonia is moving. I mean that in real world, we know that when ammonia is going through this channel, the protein also is moving. I know that we are considering the interactions between amino acids and ammonia, but we are missing that movements, and these movements may have effect on the, for example, movement of ammonia. Can we consider those movements also, movements of protein? And
I think, yeah, sorry, I think there is a mistake on the, so we are only restraining the whole center of mass of the protein. So we are not freezing the protein, or we allow the protein to move freely. The residues that interact with ammonia can interact and move. We are only restraining the whole center of mass of the protein. Okay.
Okay,
yeah,
thanks.
Yeah. Any other questions? Or did I answer your question, by the way? Do you still have a question, Delara?
No, thanks a lot.
Okay, because your hand's still up. Okay, if there are no other questions, I think I'm going to go on to the last part. So I showed here in a relatively, or Mariano and I showed here in a relatively simple system how to construct these free energy profiles. But now I'm going to show a little bit more complicated sort of system. And this is something that I've developed, that I've been working on along with one of my colleagues, Nandan Haloy. We've been working on a slightly more complicated problem, or actually, I'd say a much more complicated problem, which is understanding how do antibiotics go through proteins of the outer membrane of gram-negative bacteria. I'll give a little bit of biological background here. This is a really important problem to study because these gram-negative bacteria are highly antibiotic resistant, and it's really hard to develop antibiotics to actually treat them. And our colleague recently showed that there's a novel strategy that can be used to append, like add a certain functional group. And this can allow these antibiotics to favorably permeate into these gram-negative bacteria, where they actually show a much larger effect, a very significant effect against these bacteria. However, we wanted to study this permeation, this antibiotic permeation, using molecular dynamic simulations, and in particular enhanced sampling. But there's actually a problem in enhanced sampling. So this is something that you guys have been, you folks have been discussing in detail, is that the pathway that you're generating is actually very important. So the initial path can actually give you different answers on what the free energy is going to end up being. So for example, here, when you're doing umbrella sampling, or another method that involves seeding your free energy calculation, you need some initial pathways. So let's say you have an initial path. You want to get the free energy profile going from this state A to this state B. And so you can draw an initial path. And so people will usually draw this initial path using SMD, as has been described. But the problem is that when you do this, because there are a lot of free energy barriers that are hidden in your system, it'll be difficult to actually sample the whole space, because something here that might be in a window along this position right here wouldn't be able to actually access this position right there. So it'll get stuck here. And so you're going to miss a lot of sampling right there. And this might actually be what the antibiotic or what the substrate uses to actually get through the system. And this is actually really severe in the problem that I worked on with Nam Hawaii, where, for example, you want to see this drug going through this porin, so this protein. But the problem is that within this protein, there's a very narrow region right here. So this is analogous to this, this barrier region you can see here. That's this narrow region right there. And so because of this, and also because this antibiotic isn't just a simple system, it also, you have to consider not only the Z, but also the different angles of the molecule. So the inclination and the azimuthal, where azimuthal is, azimuthal is this angle, as you can see here, and inclination is this angle. You'll have a very limited sampling in this, this narrow region, which is referred to as the constriction region of the protein. And if you, and this can actually have some very disastrous consequences if you want to actually study any relevant properties of these systems. So for example, our collaborator, Professor Hergenrother, who is also at Illinois, had shown that if you append an NH3 group to these to 60NM, this molecule right here, that this causes these molecules to be very permeable to the gram-negative bacteria, and causes a much higher, much more significant killing of these bacteria, as can be shown with a minimal inhibitory concentration. And we wanted to study why this is, what molecular features are actually allowing this to happen. And we wanted to study this using enhanced sampling techniques. And so we used this bias exchange, which I described earlier, to identify the source of this problem. And we ran this method, and we first wanted to see if what we're running and what we're sampling makes sense. And so to do this, we constructed the free energy profile of this molecule with the amine, so with this group added, versus this molecule without the group added. And so we expect that this molecule should permeate more effectively than this one. But in our free energy, we actually saw the opposite result. We saw that this drug has a much higher free energy barrier. And so this indicates that there's something wrong with how we're initially actually seeding the umbrella sampling. So what the initial pathway is. As can be seen here, if you're sampling along one initial path, then the sampling is limited, as I said earlier. And so maybe the best pathway the antibiotic should go through might lie somewhere around here, somewhere along here, but you're not actually sampling that effectively. So our approach was to find a good initial pathway and then sample along this path. So not just using SMD to find this initial pathway. However, there's a problem. It's a sort of catch-22 problem. Is how do you get a good initial path without actually having that sampling in the first place? And so our approach is slightly different. We're using a heuristic algorithm. So we're not just using one or a few SMDs and just figuring out which SMD is the best. We sampled a bunch of pathways, and then we pick which pathway actually samples or gives the most favorable or the most likely condition pathway. And so to do this, what we do first is we first construct a series of poses of this drug. So the way that we're doing this is we're generating multiple orientations of the drug within this porin, and then replacing it in different grid points along the pore. And from doing this, we generate about 2 million poses. We remove any clashes or any ring piercings, and we minimize each pose. And so what this is doing is this is giving us a data set that we can use to identify trajectories that go through the pore until the end. And from this, we construct an energy landscape. And you want to see how is this drug actually going from this point, the extracellular space, to this point. And we determine this using another algorithm, a Monte Carlo-based pathway search algorithm, where we try to explore steps, where we try to explore the pathways that this drug is going as it goes from this space and slowly walks until it goes until the end of this pathway, or it goes until the end of this port. This can be seen here using a Monte Carlo-based approach, where each of these steps is a Monte Carlo step. And this drug has some orthogonal degrees of freedom to this z's can be seen here, which are the orientation, so the inclination of the azimuth. So those are restrained in each of these steps. And then using this, we then generate a large data set of trajectories. So each of these things you see here is actually a trajectory going from this point to the end of the point. And after doing this, we then construct a transition matrix of sorts. So and then after using this transition matrix, using all of the data, we can then determine what is the most likely pathway. So from here, this is essentially saying what pathway going from here to there is the most sampled in our system. And we find that there's two pathways can be seen here. And then using this new approach that we have, where we're constructing the initial pathways very carefully, we see that there's a much lower barrier for the aminated antibiotic, which matches the experimental results. And we then identify the permeability as well by calculating diffusion coefficient, as well as using this free energy. And we see that there's a 30-fold higher permeability for this drug, for the aminated drug, as opposed to the non-aminated drug. And in contrast, this result is the opposite of what we had found using just a pathway we had taken from SND. So my main takeaway from this is that the initial pathway that you use is actually very important when constructing these free energy profiles. So I think that was a source of questions for a lot of you people, a lot of you folks, was that how exactly are you generating these pathways, or do you have to use SND to generate them? And I guess the conclusion from the study that I did with Nambam is that this is actually very important. How you're generating those initial pathways before running the free energy can give you a lot of information about, can give you a different answer than if you run it using a poorly constructed pathway. Are there any questions about this? Does this make sense to everybody? This,
Okay, thanks, Archit. So let's take a break of 15 minutes, and then we continue with we're gonna be here answering your questions. Yeah, Mohan.
Can I ask a question from Archit?
Sure, yeah, that's perfect.
Do you have a figure that shows the pathway of your drug when you use SMD versus the Monte Carlo based?
So we do. And actually, I forgot to say that these results are actually published in a paper in Chemical Science. Those are included in a paper we have. So you can access this paper. One sec. You can access this paper here if you use this DOI. And we have that result. And what we're showing is that for the non-aminated drug, so for this drug, you see that the pathway is very similar between the SND and the this method. Whereas for this drug, we see that they're very significantly different in their orientation. Yeah, so we do see a difference using our method versus just using regular SMD.
Thank you.
And also, forgot to mention also that we have a code for this method. If anybody's interested, I can show you how it looks like very quickly. So if you you want, you can go to this website. You can look at this code for yourself. And we have a README file for how to actually set things up and the different steps for the different steps that I was talking about. This this exhaustive search for poses, this Monte Carlo-based pathway search, as well as this most likely pathway identification. If you're interested, you could start looking into this, because I think this could be helpful for a lot of you folks. Any other questions?
For this application, have you tried to see what would happen if you reverse your SMD direction? So imagine you went from the outer phase to inner phase and you got the initial pathway.
Mhm.
If you do the opposite way, you go from inner to out, would it give you the same pathway or not?
Oh, so you're saying using our method, could we go backwards and see how the pathway would look like going backwards versus going forwards?
Yeah.
I see what you're trying to say. I think we haven't tried that actually. As far as I know, I think it would be different because the way the method's working is that, one second, let me consider it here. The way the method is working is you're looking for these Monte Carlo moves when you're going from here to there. So from this position to this position. And so if you're going backwards, then it might be different because then the energy, you might have a lower energy in a different location here. So you might get different results going this way versus going that way. I mean, the question we were interested in was going this way.
Mhm.
So we didn't do that going backwards. But you could get different results using our method going one way versus another way. Did that answer your question?
Yeah, I mean, if you're using the Monte Carlo base, the probability of going from like forward and backward should be equal.
So it's not,
technically you should be able to go from the opposite direction.
So it's not really I'd say it wouldn't necessarily be a pure Monte Carlo where everything's because the idea is that you're using the Metropolis criterion to go from this position to another position. But you're also forcing it to go in one direction. So you're forcing it to go along this Z like this.
Mhm.
And so it's not that it's just exploring this using just a regular Monte Carlo, as you folks have probably are used to, where you're just allowing it to just explore different conformations anywhere. It's forcing it to go this way. So because of that, I feel like it wouldn't show the same pathway going forwards versus backwards. Also, this is stochastic, so I mean, each time you run this, it's going to be a different pathway. That's why we're running multiple of these and generating the most likely pathway using multiple trajectories. But if we're applying this with our approach, Mohammed, would you still expect this to have the same most likely pathway going this way and this way?
If your relaxation, because every time you create those poses, you relax your system. If that relaxation is reversible, yeah, you can basically calculate the work along the path that you have to calculate you basically calculate free energy, and you accept or reject based on the acceptance criteria. If that relaxation is reversible, yeah, it should be the same.
So we're not actually, so you're saying that when you make a Monte Carlo move, then you relax that system after making a move. Is that what you're trying to say?
Yeah.
So what we're doing is we're actually not doing that. We're essentially sort of bypassing that step in our method. And that's why we're calling it a Monte Carlo based pathway search. We're not really using Monte Carlo in the pure form. It's just something we're using to get a good initial path. Because what we're doing effectively is we're generating the poses at first. So we're generating these poses, we're constructing this energy landscape, and we're just walking through the energy landscape. So we're not actually walking through configurations. You can think of it as walking through just walking through grids, in a sense, you could say. You're walking through this grid, but you're using the poses to walk through the grids. So it's kind of maybe like you could say a Frankenstein sort of method, where it's not really pure Monte Carlo. Yeah, but it's Monte Carlo based because we use the ideas from there. So we're not relaxing here. What we're doing is we're getting this trajectory, we're getting the most likely pathway, and then we're relaxing the most likely pathway. But we're not actually doing any more Monte Carlo when we're relaxing the pathways. We're just using it for the price exchange that we do later on.
Because I remember from like your earlier talk is that you do some minimization to get rid of the overlap for specific poses.
Yeah, so we do minimization here in this step. Yeah, we do minimization here so that we can you get a good sort of actual point for this. It's more realistic to have a pose that maybe you might kind of clash with the protein of the drug, so you can remove that with minimization. But it's not a minimization when you're actually running Monte Carlo, because these poses are already minimized. You don't have to do that. We're not doing that again.
Thanks.
Yeah, of course. Yeah. Are there any other questions?